So this is chapter 6, part 4. So again, we're still talking about metabolism. And in this last part of the chapter, our focus is going to be on enzymes. And so enzymes, we briefly talked about enzymes back in chapter 3 when we talked about proteins, but now we're really going to hone in and focus on them. So when we think of enzymes, their major role is going to be to regulate chemical reactions. And so we can also say that they're going to be responsible for speeding up the rate of chemical reactions. And they do this, number one, in a very specific way. So every enzyme has a specific thing that it binds to, which we call the substrate. And it also does this without being changed or consumed in the process. So enzymes can be used over and over again. Okay, so again, the job of an enzyme is to regulate a chemical reaction, and they do this by speeding up the rate at which the reaction proceeds, and again, in a very specific way and without being changed or consumed in the process. And how do they do that? And so if we come over here, my second bullet point, by lowering the activation energy. Okay, so if you remember back to the first part of this chapter, we talked about this hump, right, that must be overcome in order for a reaction to proceed. That hump is called energy of activation or activation energy, and enzymes work to lower that energy requirement. And again, I'll show you a profile on the next slide. An example of an enzyme, and this is one that we'll be using in the lab or have used in the lab, catalase. And so catalase is going to be an enzyme found in the peroxisomes of our cells. It has the highest known catalytic rate of any enzyme on the planet. Its job is to facilitate the breakdown of hydrogen peroxide. Where's my pointer? So hydrogen peroxide is H2O2, and it will facilitate it breaking down into water and oxygen. And so hydrogen peroxide it's nasty. It can be toxic to our cells. It can cause tissue damage. And so this enzyme is responsible for destroying that, which is a detoxification. And so again, most enzymes are going to be proteins made of amino acids, although there are some types of RNA molecules that also display catalytic activity. So again, when we talk about enzymes, we're really usually talking about proteins, but just keep in the back of your mind that there are some RNA molecules that function as enzymes as well. They kind of a generic enzyme over here on our right, and we can see that it's folded up into the perfect three-dimensional shape. This opening right here, we're going to learn in a few, that this is called the active site, and this is where the substrate, which is shown here in red, will bind to. Okay, so whatever the enzyme is acting upon is called the substrate molecule. Okay, we'll come back to that. I wanted to quickly remind you, move my picture, of what a reaction profile looks like. And in this particular reaction profile, this is an exergonic reaction, so hopefully that makes sense to you. So if this is, the x-axis shows the progression of the reaction, so this is like time down here, and the y-axis is showing free energy. And so if we look at reactant molecules versus products, we see that products have lower free energy. And so here we state that the free energy change for a reaction of this type is going to be negative, right? So there's a decrease in free energy. And so again, this is an exergonic reaction. In red, we have the reaction profile without an enzyme. And so you can see this big hump here is the energy of activation. This is that energy bump that brings us to the transition state. This must be overcome in order to form products. Okay, and so what an enzyme does is it lowers that energy of activation. So the grayish black line shows, again, the reaction profile with an enzyme added. And so what has been done here is just the energy of activation has been decreased. The overall free energy change has not been changed. It's just the E of A, the energy of activation. Okay. Okay, just to show you the same thing for an endergonic or anabolic reaction. And so again, we've seen this. It's the same reaction profile that I just showed you, except for now it's for an endergonic reaction. So in this case, if we compare the free energy of the reactants versus the products, there is a positive change in G value. And so this type of a reaction requires ATP. However, again, the dotted red line here shows the energy of activation with an enzyme. Purple is without an enzyme. So we can see again that an enzyme will lower the energy of activation. Now notice that on this reaction profile, they're calling it a catalyst. And so oftentimes we'll use those two terms kind of interchangeably. So a biologically active enzyme is also called a biocatalyst, or just a catalyst for short. Sometimes this becomes a little bit confusing, especially in lab. We use the enzyme catalase. You can say that catalase is a catalyst. Okay, so just be careful of the wording. And you'll also notice that enzymes tend to end in ASE. So catalase ends in ASE. Amylase, we saw, that ends in ASE. Okay, so again, I put this in words here for you so that you have it. The substrate is whatever the enzyme is binding to, whatever the enzyme is acting upon, and that substrate will bind to the active site. That's the location on the enzyme of substrate binding. And so when the enzyme binds to the substrate, it forms something called the enzyme substrate complex. Okay, so the enzyme literally will, bind to its substrate. It forms what we call an intermediate complex, with the substrate. Again, I want to just restate that enzymes they show specificity. So we say that they are specific to what substrate they will bind to. So you may have heard of, lock and key. So the substrate will bind to the enzyme just like a lock and key or a hand and glove. It's a perfect fit. Okay. So again, the enzyme binds to the substrate and forms the enzyme substrate complex. That helps get to the transition state. And then the enzyme substrate complex will then release the enzyme and the products. Okay, so that's kind of a flow of how things go. Okay, so again, this is the same figure that I showed you back on one of the earlier slides. So here we've got just a generic enzyme, right? It's a protein made of amino acids that's nice and folded properly into its proper three-dimensional conformation or shape. Here is the active site, and here is its substrate. It's very specific. The substrate has to fit in here and bind. When that happens, we call this the enzyme substrate complex. Now, also when this happens, a phenomenon called induced fit occurs. And you can see this right here. So the active site, once this binds, this changes shape. Okay, so on the next slide, I've got more of a description. So again, the enzyme binds to the substrate via the active site, and this will position substrates close together to speed up chemical reactions, or it will help to contort the substrate in a way that kind of makes bonds unstable. And then this thing called induced fit happens, okay, which is just a change in shape of the enzyme. It happens after substrate binds, and again, it's going to distort chemical bonds, okay? When I think of induced fit, and you can see from the previous figure, if I can go back, yeah, what the enzyme does is it kind of like it wraps itself around its substrate. So you can kind of think of that as like a hug, okay? Again, a change in shape. And so what exactly happens when the enzyme is bound to its substrate? It could be a number of ways in which that activation energy is lowered, a number of ways in which the reaction rate is sped up. The enzyme might help to orient the reactants, perfectly towards each other so that they can react. Or, if it's a single reactant molecule, maybe the enzyme, again, puts strain on the chemical bonds, causing them to break. and for products to be formed. The enzyme, active site might also create like the perfect little microenvironment. So there might be like a little slightly different pH inside of there. So there's a number of different ways, but again, overall, enzymes lower the energy of activation and they cause reactions to proceed at a faster rate. Okay, so here is just a typical enzyme. And the substrates here are shown in orange and green. In this case, there are 2 reactant molecules or 2 substrates. That's a possibility, or it might just be 1 reactant. And so in purple here, this globular structure is the enzyme. And so we have substrates, which would be the reactant molecules. They're going to bind to the active site. That's going to form the enzyme substrate complex, and that induced fit, that hug, will occur. As the substrates are held in the active site, different things might happen, like they might be held in the right proximity or orientation. Bonds will be weakened. Eventually, what happens is products are formed. So shown here in yellow and blue are the products, and products will then be released from the active site. And notice how the enzyme is unchanged in the process. It is now available for the next reaction. So again, enzymes lower the energy of activation. They are specific to their substrates, and they do this in a way where they're not changed or consumed in the process. Okay, so I have to mention here that enzymes are going to have optimums. So there are going to be specific conditions which are going to be optimal for enzyme function. And if we get out of that window, that range of optimal, in this case, temperature or pH, the enzyme will not work. Okay? So again, an enzyme's activity can be affected, can be changed by general environmental factors like temperature and pH. There are also chemicals, right, that can also influence enzyme activity. And we'll talk about some things here a little bit later in the presentation. And again, conditions outside of the enzyme's optimal range will cause the enzyme to not function, and that might be due to denaturation. We'll talk about denaturation now. So to denature means to unfold. And so if a protein doesn't have its proper 3-dimensional shape, it can't work, right? That lock and key mechanism doesn't work. So when you hear denature, I want you to think unfold. So in this top graph, we'll quickly look at this. This shows optimal temperature for 2 different enzymes. And so the enzyme that we're looking at, okay, there's not a specific enzyme, it just says a human enzyme, and then an enzyme from a heat-tolerant bacteria. And so if we look at our human enzyme shown by the black line, we'll notice that activity is pretty low at 0 degrees Celsius, right? It's cold here. And therefore, enzyme activity at low temperatures, human enzymes at low temperatures, won't work basically because molecular motion is really slow. But as we increase temperature, right, enzyme activity starts to increase. When we get to 37 degrees Celsius around here, we hit this very high enzyme activity. And then if we go above 37, enzyme activity drops off really rapidly. Okay, so it's this part, when we get to like 40 degrees Celsius, 45, 50 degrees Celsius at a max, enzymes here will denature. The heat will cause them to unfold. Heat-tolerant bacteria, on the other hand, their enzyme activity doesn't peak until about 77 degrees Celsius. Again, slowly increasing activity and then quickly kind of dropping off. So for heat-tolerant bacteria, the optimal enzyme, window of temperature is different than a human enzyme. So it depends on where the organism is from, where the enzyme is from. That will dictate what temperature it's going to work best at. All human enzymes work best at 37 degrees Celsius. That's body temperature. So again, when you get a fever, fever is good because it's causing microorganisms in your system their enzymes to stop working, and it kills them. A high fever, though, can be dangerous. This is why. So if we get too high in our temperature range, our own enzymes will start to denature and not be able to function, and that's not good. Okay. pH, same thing. So depending on where the enzyme is from, so these are two human enzymes, and so we've got pepsin, which is present in our stomachs, and trypsin, which is present in the intestine. And so the stomach has a pretty low pH, where the intestine has a higher pH. So the stomach, we say, is acidic, and the intestine is pretty alkaline or basic. Okay, slightly alkaline or basic. So let's look at our pHs here. So for the stomach enzyme, pepsin, again, it's going to work best at a pH of a little bit under 2. If we go below that pH, if it becomes more acidic, the enzyme doesn't work. It unfolds, denatures. And the same thing if we go above that pH, the enzyme won't work because, again, it's going to unfold or denature. Now for trypsin, we have to shift our pH up here. Trypsin is going to work best at a pH of about 8. So again, an alkaline or basic environment, slightly. A pH below doesn't work. A pH above doesn't work. So again, there are these optimums or optimal ranges in which an enzyme is going to function best at. Outside of that range, the enzyme won't work. It will denature. Okay, the last part of this chapter, we're going to talk about molecular regulation of enzymes. So not temperature or pH, but these are more molecules that can regulate enzyme activity. The enzyme activity here can either be enhanced or promoted, or these molecules can reduce enzyme activity. And so the first thing we'll talk about is competitive inhibition. And so inhibition, hopefully just from the name, you're thinking this must be a decrease in activity, and that is correct. Competitive tells us that whatever molecule this is, it's going to be competing. And in this case, it's competing for the active site. Okay, so it's going to compete with the enzyme's substrate for that active site. So here's our figure. In purple, we have our enzyme. This is the active site, the spot where the substrate binds to. And here's our substrate in this teal. And so under normal conditions, the substrate will bind to the active site. The enzyme works perfectly. With competitive inhibition, a competitive inhibitor, whatever this molecule might be, binds to the active site, blocking it. And so the normal substrate cannot bind. Sometimes competitive inhibition is reversible. And so this competitive inhibitor might come off and that space might be open again. Or there are times where competitive inhibitors are irreversible. So they'll bind and that's it. That enzyme is rendered non-functional. And then there are non-competitive inhibitors, which we would also call allosteric inhibitors. And so in this case, the non-competitive inhibitor shown here in orange is going to bind to the enzyme in a place, in a site, other than the active site. We would call this position here the allosteric site. And what this does in this case is it causes a change in the shape of the enzyme so that the regular substrate can no longer bind. And so again, that would cause a decrease in enzyme activity. So whenever you hear inhibition, that is a decrease in enzyme activity. Okay, this is another figure. So on the left of the figure, it's allosteric inhibition, which we just covered. And so again, an allosteric inhibitor will bind to an allosteric site, changing the shape of an enzyme, making it inactive. There's also allosteric activators. So same idea, the molecule, the activator, whatever it is, will bind to the enzyme at an allosteric site, so a site other than the active site. That binding, again, changes the shape of the enzyme, but now instead of inhibiting function, it enhances function. And so the active site now has the proper shape to bind to its substrate. Okay, so we have competitive inhibitors, and then we have allosteric inhibitors, and then we have allosteric activators. We also have something called feedback inhibition. And so when we look at a metabolic pathway like the one here, in this pathway, we have substrate in the green, and then we have that changes to substrate A, which is an intermediate substrate, which then is changed to intermediate substrate B. And lastly, we have the little yellow square, which is the end product. Okay, so we start off with the initial substrate molecule. Here's the end product, and then we do have some intermediates. And so your cells tend to operate on a supply and demand fashion, right? They don't want to use energy, and use enzymes to make a product that isn't being used. Okay, and so what will happen is if the end product starts to build up, it will then go back usually to a very early step in the pathway. So here, right to enzyme 1, and it will bind to that enzyme, inhibiting its activity. And so we're just going to shut it down right here at enzyme 1. And so none of this is made. Okay, so your cell doesn't kind of waste time making an end product that is starting to build up inside the cell. So again, that's called feedback inhibition. Here's another example. This is a real example. The amino acid isoleucine, which is shown here, this is the end product of this pathway. And so isoleucine is an amino acid that our cells will use to build proteins. And so if isoleucine is starting to be made in excess and isn't being used, it will bind to enzyme 1 of the pathway, and it will inhibit the enzyme. And so, where's my pointer? So here we go. So here is the allosteric binding site. So isoleucine will bind here, and that's going to cause the active site to change shape, and the pathway will be halted. Okay. If isoleucine is being used, right, it will disassociate, and that enzyme will now be available. Okay, this enzyme is called threonine deaminase, and so if the active site is in the right shape, threonine will bind here. That will then change to intermediate A, enzyme 2 changes it to intermediate B, enzyme 3 changes it to intermediate C, enzyme 4 changes it to intermediate D, and then enzyme 5 is here, and isoleucine is the end product. So again, if your cells are not using isoleucine, what's the sense of going through all of these steps and using all of these other intermediates, by the way? And so what will happen is we'll shut down this pathway, okay, so that isoleucine is no longer produced. Okay, last slide. So again, enzymes can be regulated by temperature, by pH, by inhibitors, by activators, by feedback inhibition. And then our last two listed here, enzymes can be regulated by cofactors or coenzymes. So many enzymes require cofactors or coenzymes to either function, they can't function without them, or to have enhanced function, so full function. These cofactors or coenzymes come from our diets. Cofactors are going to be inorganic factors like iron, zinc, and magnesium. You've all heard of those. And then coenzymes, on the flip, will be organic molecules like vitamin C, A, B, and D. Okay, so again, some enzymes will require these things, which we call cofactors. Lastly, our cells also control enzyme activity through what we call compartmentalization. Okay, and so we've already taken a tour of the cell and we understand organelles. So again, certain enzymes may only be found in certain organelles. So here's 3 examples. So first one, enzymes used in the later stages of cellular respiration, they're not going to be found anywhere else in the cell except for in the mitochondria. And so that process only happens in there. Second one, we've actually already heard about this, but enzymes that are involved in digesting cellular debris and foreign materials, they're only going to be found within lysosomes, right? So we talked about phagocytosis and autophagy, things like that. The lysosome is involved in that, and the lysosome has digestive enzymes, and that they're going to be specifically in that compartment and really nowhere else in the cell. Last example, which we're going to be getting to in the later part of the semester, so the last module, we will talk about DNA replication. And so the enzymes involved in DNA replication are only found in the nucleus of eukaryotic cells, like ours. Okay, so again, enzymes can be compartmentalized, only found in specific locations in the cell, and that again controls their activity. So that is the end of chapter 6.